See, as you all know, that 80% of medical decisions are made on the results of IVD testing. So it is very important that IVD testing is a very critical part of the entire healthcare process. And in fact, this industry has observed a lot of changes in the way it supported the diagnosis of COVID-19. Now, we have seen that there are a lot of specific tests which has been launched recently, and which has improved the diagnostic precision of disease parameters like for COVID-19. So that has changed the mindset of people from being curative to preventive healthcare. So this has actually boosted the diagnostic services, and now everybody knows about IVD. In fact, general population also knows the value of a diagnostic testing. Earlier, diagnostic labs used to be in the basement of the hospitals, but now it is like no more the also ran it. We are IVD industry is actually the center stage of the healthcare business. Okay, so now there is a fundamental shift from a laboratory setup to now people are coming home to do the take the sample and do the testing in the laboratory. So things have changed in the last couple of years in the diagnostic industry. We are 24 years back, there was only standalone labs and hospital labs. We have not heard about big chains like ThyroCare or SRL or Metropolis and doing those beginning of 1990s. But in 1995 and 1996, ThyroCare and SRL, Ranbaxy those days, they were formed, and then other institutions were established. So right now there are totally 9 diagnostic chain labs in India, and some of them are pan-India and some of them are regional based chain labs, but they actually do bulk of the testing in the private segment. So in ensuing years, what happened was many of the small laboratories and medium laboratories, they got merged with these chain labs, and there's a bit of a consolidation in the industry, and that lab chains became bigger and bigger over the years. Mainly because it was convenient to send samples, and the cost was the cost structure changed. Specialized parameters are done in the reference big laboratories, and these chain labs used to do the routine test in their satellite labs. So this is how it was working. It was fine. It was going fine, and they used to manage the logistics of samples very well, and it was going perfectly well. However, things have changed in the present pandemic. There was a bit of a shake-up in the industry, and mainly because people couldn't, there was a bit of a transportation issue, and people wanted results immediately. So especially for COVID. Now, so this has made a small and medium sized lab to wake up and sort of look at their infrastructure and ensure that they have enough tools to give these results what the patient needs quickly. So that is how the present pandemic has changed all that, and we see a shift towards specialized testing being done in smaller labs closer to patients. So basically, the point here is the labs needed to be close to the patient, because patients couldn't go far distances to get their samples tested. So that is how we see a lot of smaller laboratories upgrading themselves and ensuring that all kind of parameters are being done in those laboratories. So just to give you a perspective, we have the IVD industry is about 9,300 crores, about 10,000 crore market, and out of that 47% is small and medium sized labs. So there's a significant infrastructure there to do the testing. The lab chains are about, if you see the graphics, it's about hospital labs are 37%, and small and medium standalone labs are 47%. Balance is chain labs. So and this small and medium sized labs are going to grow further because of the infrastructure already built in, and that will bring in more samples, so it will grow. So as I said, most of the workload earlier was consolidated into 9 major chain labs, but because of travel restrictions pandemic, it has gone to smaller samples, go to smaller standalone labs too now. So we have seen actually in the last a couple of years that some of the labs, they never thought about molecular testing earlier before pandemic, but as soon as this COVID hit, they these labs then upgraded the infrastructure, and they brought in nucleic acid separation instrument, PCR, RT-PCR instruments. So this is a new thing which they incorporated in their laboratory, and we see at a 400% increase in the number of labs adopting molecular testing, which was unheard of earlier. So this is something, which was the need of the hour, and this was done by the small and medium-sized labs. So if you see, we have a product called Aquila from Thermo Fisher, which is a small POCT kind of RT-PCR kit. That has been a new introduction, and it's an innovative product wherein you get the result within 30 minutes. So these kind of products have become mainstream product now in the country. And continuing with that, I would like to say that the market for POCT is really well established in the market. Earlier, it was like not very structured kind of adoption in the POCT for the POCT product, but now most of the medium-sized labs have adopted POCT, and especially POCT means you do tests which are required for COVID, like IL-6 or ferritin or D-dimer, CRP. Those tests you can do on this POCT, and most of the samples which were coming last 2 years for mainly for these tests. Of course, apart from this, you still can do tests like like you can do CKMB, you can do NT-proBNP, troponin I, then hormones, T3, T4, TSH, LSFH, prolactin. All these tests you can do on getting 1100 like system. So this product has been adopted in all the nursing homes, all the standalone labs. So this was a need of the hour, and people bought these machines to ensure that they give the report within half an hour or 1 hour's time. So this was a big major change. Otherwise, earlier what used to happen was those samples were collected and they used to go to they used to be sent to a reference lab or chain labs, and then you'll get the result next day. But here now, what happened was they wanted the results to be given immediately, and that was possible by this POCT point of care testing instruments. So this was a big change which we saw. Earlier it was people are adopting it, but in a very slow pace. But now, because of COVID, this has totally changed. And additionally, we have seen that many of the laboratories, they used to outsource their thyroid or hormonal assays to big chain labs. So, but because of COVID, they started investing on a smaller immunoassay chemiluminescence analyzers, and they are now doing all the tests themselves. That is a big change what we have seen. And because we have noticed that we also have a product called Meglumi 800 machine from for CLIA based assays, and the uptake of this product has been quite good in the last few months. So that means smaller labs are equipping themselves with these machines, so that they don't have to send their samples to far distances, and they can do the result and send the result out quickly. So this is a major change what we have seen. Then many, as I said earlier, many labs have gone for nucleic acid testing because that was they were getting a lot of samples for that during COVID, and they found there are some instruments which are like very small machines and have the same capability of a big RT-PCR analyzer. So those systems have been procured by this lab, and now all these labs are fully equipped with these nucleic acid detection systems. Now, right now it is only for the COVID, but then once COVID goes, then they can be adapted to other infectious diseases as well. So all this, now we have seen that even if you see an example which I want to tell is we have there has been only few labs throughout the country doing prenatal testing, or they were sent to some reference laboratories for doing prenatal testing. But now we see that the demand for this product has gone up, and many people would like to do it at in their own labs, and that is something which we realize that how the pandemic can change the thinking, and people start adopting this technology in their own labs. So, which is a good thing, and so all this proves that small and medium labs are the way forward, and they'll only expand further. Even though COVID will go, but then they will expand further because they have the infrastructure. And there's integration of new biomarkers like IL-6. Very people used to do earlier. Now many of the labs can have capability of doing interleukin 6 and PCT and those tests for COVID, and the availability of the biomarker tools present the new opportunity for this new smaller labs. Now earlier smaller labs were doing just glucose or basic tests, glucose or lipids or some kidney profile and some hematology tests. But now because of this adoption of this new technology, they can report all the results in a very short time span of time. So the most importantly, by doing the testing closer to the patient, which is very, very critical, especially in the pandemic, the issue of sample deterioration is also mitigated. There's no problem in sample deterioration. And finally, I'd like to say that GenWorks has all the product can provide full solution to equip smaller and medium-sized IVD labs with necessary tools to assay all routine and specialized parameters. So, of course, the chain labs will expand. That is no question about it. But because the number of testing in the country has gone up, all the small and medium labs are getting enough sample load to do these tests. And since most of the patients are paying patients, like they pay for the test, it is always they want the test to be done quickly, and they get the report for their further use in a short period of time. So that is my submission. Thank you very much. Thank you.
So if you see today's lab system, here the patients are coming with burden of disease. Then we use our intelligence, discipline, and creativity. It's very important to use all the three inside our lab. And then patients are going out with reports. So either they are happy or satisfied, or with our reports, clinicians are our customers, then executives in executive health checkup. So they are either happy, or sometimes you may get called like Barnali, what TSH report you have given. So this is not correlating with the clinical scenario. And if you see this person, this may be you or me, being an ABL assessor and College of American Pathologist inspector, we do sample audit for 2 days or 1 day. And based on that, we say, oh, there is no deficiency, or there are so many non-compliances or deficiencies. So this is our today's lab system. And so why there are so much variation? So do you think this is the reason? We know sample integrity caused a lot of variation, and during my assessment, I have seen samples transported in different modes of transport, although I have not seen yet the elephant, but yes, samples are transported. So do you think the variation in TSH reports due to sample integrity? And so outline of my today's talk, standardization and harmonization, and in lab medicine, and how we can harmonization of different phases like pre-analytical or examination phases, then analytical and examination phase, and post-analytical or post-examination phase. So pre-analytical and analytical and post-analytical phase harmonization is the need of the hour, and the quality indicators of harmonizations and who is responsible for harmonization and test standardization are we nearly there yet, and few examples like TSH and few others. So if we say standardization, so where there is a primary reference measurement procedure, so this is called standardization, and harmonization where there is no primary reference measurement procedure, we have secondary reference measurement procedure. So we do it by all-purpose streaming, that is called harmonization. So standardization where there is primary reference measurement procedure, then secondary reference measurement procedure, manufacturer reference measurement procedure, and end users routine measurement procedure. So then we can standardize the test. That means standardization is full metrological traceability. And where there is a partial metrological traceability, like there is no primary reference measurement procedure, we only have manufacturer master measurement procedure or end users routine measurement procedure. So then we can't standardize, we can only harmonize. So for that, we need all-purpose stream domain, and based on that, we can harmonize that method or that procedure. So for example, thyroid function test, T3, T4, total T3, total T4, and free T3, free T4, small molecules, PO standards, and pure primary reference measurement procedure available. So capable of being measured by isotope dilution, mass spec, and FT3, FT4 after equilibrium dialysis. So standardization is possible for T3, T4, and FT3, FT4. And for TSH, it is a complex glycoprotein, different isoforms. Then primary reference measurement procedure is not there. And because of that, all-purpose stream domain represents the best target. So their harmonization possible. So how do our process in our lab look? So producing to target value means we need to change our habit. So that is actually the role of standardization and harmonization. So harmonization, according to CLSI, the process of recognizing, understanding, and explaining difference while taking steps to achieve uniformity of results, or at minimum a means of conversion of results such that different groups can use the data obtained from assays interchangeably. And coming to standardization, there is full metrological traceability by ISO 17511. Standardization involves more stringent, higher order reference materials and measurement procedures than harmonization. So why should we harmonize? Because of patient safety. We just celebrated Patient Safety Day. So patient safety, patient empowerment, and public confidence. Patient safety by accreditations, consolidation, and networking is important. Patient empowerment, like clinical guidelines, informatics, public confidence, clinical governance, and electronic patient records. So this all we should harmonize. And if we talk about total testing process or brain-to-brain loop or Ludenberg loop, so if you see this is patient goes to clinicians, then need for lab medicine, then pre-analytical, analytical, post-analytical steps in the lab medicine phases. And then it again, the report goes to doctor, and then it goes to patient. So this is the total testing process. So we need to harmonize the total testing process. So standardize or harmonize lab protocols, we need to do test requesting, sample handling, reporting pattern. So this is done at local level, lab director or lab consultant should do that. Then lab parameters like test names and units, reference intervals, critical values that we need to harmonize. So that can be done at local or national level, like at the local lab level, in-house, or national level, like NABL. Those bodies can harmonize those procedures. Then lab methods like technology, traceability, commutability. So this is done at local and international level, like IFCC plays very significant role. Then CLSI and different other organizations like AACC, IFCC and all. So this standardization and harmonization, we can harmonize the test. So coming to specimen collection, so there are variability in specimen collection. So this is a major source of pre-analytical error. So initiatives to improve and harmonize specimen collection, guidelines on patient preparations, including time of collection, then international standards for phlebotomy, training, competency, uniformity of color coding, and use of blood tubes, lab practice guidelines for users. So reducing between method variability, how? Comparable results by monitoring like consistent performance maintained via proficiency testing or external quality assurance, adjustment of the calibrators and result, then designing calibration and traceability to common reference system, and then standardization and harmonization. So this, if you see method standardization and harmonization according to ISO 17511, standardization is possible where there is a primary reference measurement procedure. So electrolytes, glucose, creatinines, enzymes, we can standardize. But like some proteins, tumor markers, then like TSH, we can't standardize. Then we can only harmonize those tests. So nomenclatures, units, report formats like SI units, or for example, HbA1c, like we need to do the units harmonization. Like should we report in NGSP percentage or millimole per mole? So that is like, units and report format harmonization. So reference interval is very important, and there is excellent CLSI guideline for reference interval. So it should be gender, age, and ethnic specific reference interval. So there is IFCC committee for reference interval and decision limit. So who are the stakeholders in harmonization and standardization? Diagnosticians from lab, then diagnostic manufacturers, IVD industry, regulators like accreditation bodies, then patients, clinicians, and lab staff. So all stakeholders can play a very important role in reducing variability in patients' results. So now I'll come to IFCC Standardization Committee on Thyroid Function Test efforts. So like there are attempts to harmonize commercially available TSH immunoassays. So this began at phase 1 trial with method comparison using sample from presumably healthy individuals, and then in which we showed that recalibration using all-purpose trimmed mean significantly increased the agreement of commercially available assays. Then phase 2, allowing the manufacturers to individually adjust their own calibrators using the APTM or all-purpose trimmed mean from another method comparison with a similar panel of thyroid samples. And phase 3, recalibration to the all-purpose trimmed mean was similarly successful using samples from patients with thyroid disease. So there is overall excellent correlation of most of the immunoassay results to the all-purpose trimmed mean in patients, and with both hypo and hypothyroidism led the committee to conclude that the assay measured TSH in equimolar fashion regardless of differences in glycosylation. And phase 4, we attempted to show that our approach for recalibration may allow manufacturers to have more uniform reference interval in future. So this is our paper in clinical chemistry, harmonizing TSH measurement, and this an adoption of more uniform reference interval, and another paper for standardization of free thyroxine measurements and adoption of a more uniform reference interval. So we have shown that after recalibrations, if you see that variation decreased between different IVD manufacturers, different IVD assays. Here, I believe in the philosophical lives of Albert Einstein. Life is like riding bicycle. To keep your balance, you must keep moving. So how we should harmonize my laboratory TSH results? I'm sitting in a lab in India. How I can harmonize? And so if we see these two wheels of this lab life cycle, so one is the wheel of quality, another is wheel of cost. Wheel of quality depends on variation, depends on accuracy, precision, reference interval establishment or cutoff, and wheel of cost depends on three magic M's: men, machine, and material. So validation or verification play a very important role in harmonizing between these two wheels. Let's see how. So here I'll give our lab example. We have run samples by both new and comparative methods to evaluate bias. So we have run 20 samples in the analytical measurement range, and then we have verified the precision within lab precision and within run precision. We have verified 5 different days, 5 runs, so 25 samples altogether of each of the quality control sera. Then we verified LOB and LOD, limit of blank and limit of detections, and 20 blanks or zero calibrators or low patient samples by verifying that. Then linearity verification, we call that as a range that can be reported without dilution or pre-treatment of samples. That is called analytical measurement range, or AMR, and extended measuring interval, or EMI, according to CLSI EP34 range that can be reported with dilution or pre-treatment of samples. So this is very important when we report tumor marker. Then if we need to know what is the AMR and what is the CRR, or extended measuring interval. So for all the immunoassays and biochemistry tests, we should not go beyond the EMI or extended measuring interval. And lot-to-lot verification, according to CLSI EP26A, verify patient samples of 2 key decision concentration 2 or 3, and you can customize that at the local level. And along with the patient samples, you verify the QC samples and to assess the performance at clinically important concentrations. So coming to reference interval verification, if you see, we can take adopt the reference interval according to CLSI C28 guidelines, or manufacturer suggested reference lab, reference textbook, articles, or pack insert.
Now, let me show you a few cases. These are the real life cases which happened in our hospital, and what actually prompted us to look at point of care testing in COVID. This is a first case, which is a 32-year-old lady who is a case of acute leukemia. She had acute myeloid leukemia and was posted for stem cell transplantation or bone marrow transplantation. That's what is otherwise known. The patient was absolutely stable. However, the tests which had been done on this patient had suggested that this patient needs a transplant and needs to be getting it done as quickly as possible. Now, the biggest challenge was we were awaiting a COVID test. Now, the moment we wait for a COVID test to happen, we have to actually have some activity which needs to be done as quickly as we can. Another case wherein in one of our facilities in Mumbai, we had a crisis of the nursing staff. Again, you know how COVID was. So we had to make sure that we depute staff to the facility there. And of course, to catch the flight, you again need a COVID test to travel. And again, it's not an easy thing to do, because you are again confused as to how you will get your test done quickly for such people. Now, again, the COVID test was awaited. Another case scenario wherein we had a 54-year-old man, who had obstruction of intestine and was posted for emergency surgery. Now, again, the issue was what to do. How can we take the patient up for surgery? Because again, the COVID test was awaited. So in all these circumstances, if you look at the whole scenario, the question is how quickly can we get our test done so that we are able to give a quick answer? Again, another patient in whom we had a case of what we call a mantle cell lymphoma, a kind of a cancer which needs to, be treated as quickly as possible because again, the patient was having symptoms which needed to be alleviated. Again, the question was what to do because we were waiting a COVID test. So when we look at COVID-19 diagnosis today, there are lots of challenges. Of course, collection of the sample itself is a big challenge, and then subsequently transporting the sample to the lab. And then eventually within the lab, we should have an adequate number of samples in the batch to go ahead with the testing part of it. So if we look at the issues in all these cases, which I just gave you examples of, when we look at it from the patient's perspective or a perspective who's treating the patient or from the laboratory perspective, there are three important aspects which all of us are looking for. One, we want to test as quickly as possible. We want it to be reliable and we want it to be cost effective. These are the three important aspects which are there of paramount importance in the minds of all of these three people, whether it's a patient, a clinician, or the lab physician. And amongst all of them, rapidity or the quick turnaround time happens to be one of the most important aspects that we are looking at, especially in the circumstances that we were placed in. So if we look at other aspects of COVID, which COVID put the hospitals into, was that there were lots of patients who were not having COVID and they were having other issues, like they would have, just as the way I showed you, cancers or transplants and stuff like that. And now, therefore, you needed to take a call of making sure that you don't mix these patients with positive cases, because obviously COVID would then give them more problems than solutions. Again, you would want to eliminate some asymptomatic cases. You would want to make sure that the time taken for waiting for the test should be as minimal as possible. And of course, this would also ensure that you could schedule your surgeries or other procedures appropriately and would help you to have appropriate time and operational management. So all these aspects were important because of the operational issues that you were looking for, and therefore having some solution to this was very important. Now, what was happening in majority of the places, across the globe, in fact, and not only restricted to our country or to my hospital, we were having these COVID-19 patients, and these COVID-19 patients obviously were immediately put into a quarantine ward. And then from the quarantine ward, they would be tested, and depending upon where they would be put in buffer zones. And then definitely, then one would have to define whether they are having COVID or not having COVID, and eventually they would be getting them out. Parallelly, there will be patients who may or may not have COVID, or so-called suspected COVID patients. Now, they would probably be in a clean area. And if you have a clean area where you had non-COVID patients also, they would, interact with each other and probably transmit COVID to one another. Obviously, you needed to do something for the buffer zones. And therefore, what we did in our hospital was from the OPD or the emergency area, we would have a buffer ward where all of these patients will go. We'll test them for COVID. Eventually, the patients who would not have COVID will go to the non-COVID zone, and the ones who would go to the COVID zone. Now, it would seem very simple. This would be a very good thing to do, but at the same time, two major issues. One, you had to have some beds which were getting occupied for a long time before you get the results out. And second important part, despite the fact there would have been some patients, who you would think they are non-COVID, but if they're asymptomatic, they would actually be harboring COVID, and they would probably give it to the other patients as well. Now, in these circumstances, you wanted to make sure that you do something which would give your results as quickly as possible. And that's where the operational issues started coming up. You had patients who were not happy with the whole circumstance. You were not able to give them the logistical system so that they could, be taken care of appropriately. And obviously it led to a lot of discontentment amongst patients and of course your staff as well to have, to handle this. Now, in our quest to decide, to get a COVID test, which was quick, reliable, and cost effective, we started embarking on a journey and tried to see what test could help us to make it sure that we are able to give a quicker result so that the patients can be taken care of appropriately. And guess what? The person who came to our rescue at that point of time was our ex-Prime Minister of the President of the United States, Mr. Donald Trump, who actually unveiled one of the methodologies of doing testing on a point of care basis to give quicker results. This is what we adopted, and we call it the cartridge-based nucleic acid testing systems for COVID-19. Fortunately for us, this was a system which was available in India at that point of time, and both ICMR and Drug Controller General of India had approved it, and it gave us a result in as less as 13 minutes and was comparable to RT-PCR, and therefore was adopted universally across the country. So when we use this technology, in this particular technology, the platform that we used was ID Now, and in this ID Now platform, we use what is called isothermal technique of nicking enzyme amplification reaction, and then using the recombinase polymerase reaction to get the results out as quickly as possible. A specific gene of the COVID-19 was used in this particular case. The gene which was in picture in this case was the RDRP, and the conserved regions of this particular gene, which were highly conserved regions, were taken up, and it worked specifically at 56 degrees temperature, and we ensured that the results are given quickly. Just to give you a brief insight into how nicking enzyme amplification reaction works. So you have these two distinct enzymes. One is the enzyme which is causing the nicking or cutting of the DNA template. And then you will also have the enzyme, which is, the polymerase, which is ensuring that you are able to actually propagate the reaction. And therefore, if you see this, you nick and you propagate, and you nick and you propagate, and that's what keeps happening, and you're able to get multiple copies. And with the number of copies that you specifically require to actually give the results, you are able to quickly give the results out, give results as quickly as possible. So if you look at it, the good thing was that you were able to integrate this particular system with the laboratory information system, which was able to give the results into the patient record as quickly as possible. So therefore, perhaps CBNAT testing, was a changing event in the circumstances that we placed in our organization. And how did it help us? It helped us significantly. We were actually in this kind of a concept where we had patients coming in from the OPD and the emergency areas, moving into a buffer ward, waiting for almost 12 to 24 hours. Then once the results would come out, they would go into either COVID or the non-COVID zone. Now, what happened the moment we adopted this CBNAT technique? This is how the whole thought process completely changed. Whether for COVID patients or for non-COVID patients, we went ahead and did this CBNAT-based technique, and we figured out whether it was a positive or a negative case without making them get into a buffer zone. And at the same time, we were able to ensure that they would be transmitted to the respective areas very clearly. If somebody was positive, he would be sent to the COVID care. If they had a flu-like symptoms, if they were negative, they were sent for home care. On a non-COVID front, again, the positive and the negative would be adopted accordingly. So you will have cases which were positive, they would go to the COVID zone, and the ones which would be negative would be sent to the non-COVID zone. Now, this helped us a lot because it ensured not only the patient satisfaction, but it also ensured that our buffer zones are taken care of. So operationally, if we look at it, the buffer zones got eliminated. The waiting time for the tests got reduced significantly. We could schedule surgeries and other procedures up to the mark, and there was appropriate time in operational management. So this helped us significantly, in terms of operations of the organization. And there was a marked improvement in the patient satisfaction. And if we look at the CBNAT, this is how CBNAT picked it up. CBNAT also picked it up significantly. So in fact, if you look at the percentage positivities, the percentage positivity picked up by CBNAT was far higher compared to what you were able to pick up on a conventional RT-PCR. Clearly indicating that we were dealing with something which was important. Now, just to give you an idea, this is the month of April 2021. As you can see that there is a significant peaking at that point of time, not only in terms of total cases or daily new cases or active cases, even deaths at that point of time were pretty significant. Fortunately for us, things have started changing. There are increased number of recoveries also which are happening at that same point of time. Of course, the deaths are also coming down. And this is our current state where we are in our country as on August 2021. We have seen that the wave has come down significantly. It's almost coming down towards the baseline. But what is very important to realize is we still have a very big and a prominent case workload, which is happening in the state of Kerala and of course some coastal areas across and some parts of Northeast as well. So we cannot just leave the guard. We have to make sure that we have availability of testing quickly and we're able to give the results up as quickly as we can.